Hey everyone, I hope you're all doing great. This is Laboratory Stuff, and today we will be talking about CBC or Complete Blood Count. First, let's talk about blood and blood components. Blood consists of 4 components: plasma, RBCs, white BCs, and platelets. Plasma accounts for 55% of the human blood. It consists mostly of water, and it carries blood cells, platelets, proteins, ions, nutrients, hormones, and wastes. RBCs, it carries oxygen from the lungs to the rest of the body, and carbon dioxide from the rest of the body to the lungs. White BCs, it produce antibodies and consume pathogens by phagocytosis. Platelets maintain homeostasis and repair tissue injuries. CBC, Complete Blood Count. It's probably the most requested lab test, and it's used as a checkup routine. It works under Coulter principle electrical impedance, and it's basically a group of tests which are RBCS count, hematocrit, hemoglobin, MCV, MCH, MCHC, white BCS count, RDW, white BCS differential, platelets count, and MPV. Today in this video, we probably will be talking about RBC stuff like RBCS count, hematocrit, hemoglobin, MCV, MCH, MCHC, and RDW, and leave white BCs and platelets for another part of the video. RBCS count. It measures the number of RBCs in millions in a microliter of whole blood. RBCS count is used in calculating RBCs indices, which we will be talking about in a minute, and it helps in the diagnosis of anemia and polycythemia. Normal range for men is from 4.5 to 6.5 million beer microliter, and for females from 4 to 5.5 million beer microliter. But sometimes when you have high levels of white BCs, that could lead to a false high RBCs count, and if you have agglutination or Rolexes, this will lead to a false low RBCs count. Hematocrit. It measures percentage by volume of packed RBCs in a whole blood sample. Normal range for males from 40% to 54%, and for females from 36% to 47%. But what does that mean? Low hematocrit suggests anemia or dilution or massive blood loss. Why anemia? Because in anemia you have a low RBC count, so you have a low hematocrit, and in dilution you have a high plasma. High hematocrit indicates polycythemia and dehydration. As well, polycythemia means high RBCs count, and dehydration means less plasma. Hemoglobin. It measures the amount of hemoglobin found in a deciliter of whole blood. Normal ranges for male, it's from 13 to 18 grams per deciliter, and for females from 12 to 16 grams per deciliter. MCV, mean cell volume. MCV, MCH, and MCHC are called RBCs indices because they are measured from equations using other known indicators. MCV is the ratio of hematocrit to RBCs count. It expresses the average size of erythrocytes, so it shows if the cell is microcytic, normocytic, or macrocytic. The normal range is from 80 to 100 femtoliter, and MCV can be measured using this equation. Low MCV indicates anemia caused by iron deficiency anemia or thalassemia, and high MCV indicates anemia caused by folic acid or B12 deficiency. MCH, mean cell hemoglobin. The hemoglobin to RBC ratio. It gives the weight of hemoglobin in an average erythrocyte. The normal range is from 27 to 32 picogram per cell. and it also can be measured through this equation. MCHC, mean cell hemoglobin concentration, the ratio of hemoglobin weight to hematocrit. It defines the concentration of hemoglobin in 100 milliliter of bagged RBCs. It shows if the cell is hypochromic or anormochromic. The normal range is from 30 to 35 gram per deciliter, and it can be measured through this equation. RDW, red cell distribution width. It's a measurement of the range of variation of RBCs. Normal range is from 11.5% to 14.5%, and it can be measured through this equation. RDW is normal in genetic anemia like thalassemia, but it's elevated in nutritional anemia like iron deficiency anemia and B12 or folate deficiency, and you can differentiate between these two by MCV. Thank you all for watching, and hope you found that useful and enjoyable.